Well, it's very nice to be here at this very exciting meeting. The aim of my presentation is to draw together some of the different ideas that we've had, particularly how signals from the digestive system are integrated in the brain with the inputs produced by food, by the taste and so on, that produces reward. And I'm going to therefore be considering if these are in balance properly, and if that is something to do with what causes obesity, a possible imbalance in that system. Now, the person who has particularly contributed to some of the work I'll be talking about today is Fabian Grabenhorst, and some of the approach is described in this recent book that's just come out about emotion and decision-making. And you may wonder why Adam and Eve are here. And the reason for that is that you could think of that in some societies as being the first emotion and the first big decision. Here's the sort of framework that I want to present to you for what's going on. So we have sensory factors, taste, smell, texture, and sight, which are coming into the brain here. And through these, we have effects of variety, sensory-specific satiety, palatability, just how good the flavor is, the food concentration, which we've heard about, the energy density, portion size, and ready availability. So all of those are the sort of inputs, the sensory inputs, that are going to be converted into the brain into reward signals. And I want to consider exactly where that is done and how it's influenced by then these satiety signals that switch off the feeding. What are the rules there? And it turns out that the brain has cognitive effects, such as what you think about a food, advertising, coming down into our reward systems. And it directly modulates the reward representation produced by food. And I'm going to show you evidence for that. Then the output of this system has to produce, for example, eating and autonomic outputs and endocrine effects. And this leads to the very simple hypothesis that there's some sort of imbalance here, that in obesity we have enhanced sensory-produced rewards produced by all of these factors, which override satiety signals, which haven't, of course, changed. They've been genetically set over tens of thousands of years, whereas many of these factors have changed a lot in the last 30 years. So that's the hypothesis. This is just a little reminder about some of the parts of the brain crucial for food intake. This is the back of the brain, the visual cortex, and the front of the brain. Your eyes would be about here, so that's orbitofrontal cortex. And orbitofrontal cortex is very important in food intake control, as is amygdala in the temporal lobe, because they receive inputs from the primary taste cortex here in the insula, from the primary olfactory cortex, but also from visual cortex about the sight of food, and also from somatosensory cortex about food texture. So it turns out that this is a very important site of convergence, as is the amygdala, for all the stimuli which are going to tell us about food reward. And this is a schematic of how the system is wired up in primates that helps us to understand how satiety signals, which I'm labeling here with a hunger neuron, come up into the lateral hypothalamus and into the orbitofrontal cortex and probably the amygdala to influence the effects of all of these stimuli, the vision, the taste, the smell, and the touch of food. Now, for example, the taste pathways come through the brainstem in primates necessarily to the taste thalamus and then to the primary taste cortex, then into orbitofrontal cortex. So this region is mainly unimodal, the primary taste cortex. Neurons do not respond there to odors. Olfactory inputs come into the primary olfactory cortex and then reach these structures, orbitofrontal cortex and amygdala. So does touch. So does vision from inferior temporal. Now it turns out, and this is going to be one of my themes, that everything in this tier, the what tier, primary taste cortex, etc., simply tells you what the stimulus is, independently of how nice it is. So in humans and in monkeys, what a food is clearly separated from how nice it is. It's only when we get on to places like orbitofrontal cortex and amygdala that then we find effects of hunger influencing the effects of food. Therefore, that's where reward is being represented in places like orbitofrontal cortex and amygdala. So there's a very clear distinction. And it turns out, as you'll see, that we can modulate how much these different systems are responding by paying attention, for example, to pleasantness. We turn on orbitofrontal cortex relative to the taste insula. So we get these top-down influences from cognitive parts of the brain that can influence the way in which even our taste system is processing information. And that's going to be very important in determining how you respond in any situation, whether it's in a marketing situation or in a food research laboratory. The instructions that you give will have major influences on activity in orbitofrontal cortex and primary taste cortex, as I will show you. So that's the sort of anatomical framework. Now, just very quickly, a little bit of some of the hard data. So this is a single brain cell in the macaque orbitofrontal cortex. At time 0, a taste goes into the mouth, sweet. These little spikes are the neuron responding. You can see how selective these neurons can be. This taste neuron in the secondary taste cortex does not respond to salt or sour or bitter, quinine. Doesn't even respond much to fruit juice. Notice. So it can tell the difference between sweet and fruit juice, and it doesn't respond to water. So that's what we have studied a lot. Studying this sort of neuron, we discovered sensory-specific satiety, in fact, first in the lateral hypothalamus. But this is the same experiment. So have a single neuron in the orbitofrontal cortex, and it goes up from baseline of a few spikes a second to 20 spikes a second when the monkey is given glucose. As we satiate the monkey with glucose, feeding 50 mils up to 250 mils of glucose, then the response of this neuron gradually decreases to 0. And so does the monkey's feeding behavior. So the reward value here goes from high, +2, he's accepting it, to down here where he's rejecting it. And the neuron parallels the change in reward value. And as a matter of fact, if you put an electrode into this region, an animal will self-stimulate electrically to activate this set of neurons. Now, sensory-specific satiety is here because if you test the same neuron with fruit juice, BJ, black currant juice, the neuron is still responding at the end of the satiety run. So that's actually how we did discover sensory-specific satiety. It became very obvious when we were counting spikes per second, and we went on to do lots of experiments in humans to test and show that the same thing works in humans. Now, the next point I want to make is back to perception is separate from hedonics. So this is now back to primary taste cortex. And here's the same satiety experiment. Feed to satiety with glucose, and the neuron in this bit of primary taste cortex does not change its activity when you feed glucose to satiety in response to the taste of glucose. So that's the evidence that reward is not represented in the primary taste cortex. Now, one of the most important things that we've discovered about the orbitofrontal cortex is that it's like a reward antenna on the world. It's telling you all about the specific details of foods by the different responses of different neurons. So here are two cases in point. This is one neuron. This is another neuron. Here's the firing rate in spikes per second. And this neuron's responding a bit to glucose and to fruit juice, not to sodium and monosodium glutamate. But have a look up here. It does respond to viscosity. This is carboxymethylcellulose getting thicker. It does not respond to fat in the mouth, and it does respond to capsaicin. This neuron, on the other hand, has no taste input. It does have a viscosity input. It does respond to fat in the mouth, and it does not respond to capsaicin. So that's how you're able to compute sensory-specific satiety, by having neurons that are fairly specifically tuned to the reward value of different foods. All you need to do is let adaptation take place, and your liking for that food in this reward system goes down, but remains high for other foods. There's an important little note here relevant to dopamine. So reward representation as a goal for action must be specific for a particular reward. We have to know that we're going after, say, a food versus water versus something else. But it even has to be more specific than just food. We need to know which food it is so that we can get these effects of variety on food intake. And as far as we know, nothing like that is true of dopamine. So dopamine is not transmitting a reward signal in any simple sense that's relevant to us selecting something like a food. So it may be modulating our food reward system, but it's not carrying the main specific food reward signal. That is what's represented in the orbitofrontal cortex. Now, here's an interesting discovery that we made, which I think is very relevant to the development of new foods that may be highly palatable, but may be non-nutritive. So here's another neuron in primate orbitofrontal cortex. This one has no taste input at all. It responds with a high firing rate to cream in the mouth, fat. It also responds to vegetable oil and trialine. So it responds to natural fats. But this sort of neuron responds rather well to paraffin oil, which is pure hydrocarbon, and to silicone oil, which of course has no fat in it. So this is a fat texture sensing system, which we believe is important for the main mouthfeel that you produce for fat in the mouth. These neurons do not respond to fatty acids, such as lauric acid and linoleic. A few other neurons do respond to linoleic acid or lauric acid, so fatty acids are probably being transduced, but they're not accounting for these responses to this wide range of fats. Now, the implication is, if the food industry could develop new substances which produce this texture effect, but with no energy density, then you might be on the way to producing something that would be palatable, but which could be incorporated into a healthy food. Now, because I think these neurons are quite interesting, I'm just going to show you a little bit more information about them. They are something to do with reward, because here's a neuron that increases the cream in the mouth when the monkey's hungry. But when you satiate the monkey on cream, the neuron stops responding to the cream, but still responds perfectly well to glucose after satiety with cream. So this is showing sensory-specific satiety for fat, as opposed to what hasn't been eaten in the meal, glucose. So these are reward neurons for the texture of fat. Now, just to draw out what's being transduced, there are lots of different types of neuron in the orbitofrontal cortex. This particular neuron, with its firing rate up here, is going as a simple function of viscosity. So this is a carboxymethylcellulose series, getting up to about the thickness of toothpaste, and this neuron reflects that beautifully, and the responses to all these oils fit exactly along the viscosity curve. So that is a viscosity neuron. Viscosity is being sensed. But the point is, there are lots of different sorts of neuron. So this, in contrast, is one of the fat-sensitive neurons, fat texture-sensitive, which does not respond to the carboxymethylcellulose at any viscosity, but has a high firing rate to all of these oils, which is actually essentially independent of their viscosity, which is the number that's down there in centipoise. So that's to make it clear to you that there is a signal in the primate orbitofrontal cortex about fat reward in the mouth that's independent of some other texture signal, such as viscosity. So there is something that the food industry might be interested in here for developing new classes of food. And now a quick summary of what we know from the monkey about food reward in the orbitofrontal cortex. Taste reward is there, so is olfactory reward, which I haven't talked about. I have talked a bit about texture reward. Visual reward is present, and neurons learn in one trial to respond to the sight of a food, and in one trial they can unlearn that in primates. The important point is it's a neuronal representation of stimulus value, how rewarding the stimulus is. It's nothing to do with behavioral actions being taken. So this is like your hedonic window on the world. It's telling you about how nice things are, but not what to do about them. Okay, now to what extent then do we find similar things true in humans? Well, this is one of our early human neuroimaging studies. This is a coronal section through orbitofrontal cortex in a sensory-specific satiety experiment. So the humans could have tomato and chocolate to taste. Then on one day, they were fed to satiety with chocolate, and we found that the bold signal, the signal we measure in this location of the orbitofrontal cortex, is high before they've been satiated, and as they're satiated, the bold signal goes right down, as does their pleasantness rating. So the pleasantness rating is correlated with the activations in this bit of the brain. And this happens for whichever food you've eaten in the meal. So if you ate tomato on another day, then you would see this effect go down for the tomato, but no change for the chocolate. So sensory-specific satiety in the human orbitofrontal cortex. Oral fat texture, too, in places like human orbitofrontal cortex, but also the next region on, tertiary taste cortex, which is this cortex just in front of the corpus callosum, anterior cingulate cortex. So this neuron responds, this activation in humans occurs to fat in the mouth and much less to carboxymethylcellulose. And interestingly, this region and the orbitofrontal cortex, which also has this type of signal, both project to the ventral striatum, which also shows this response to pleasant fat in the mouth. Indeed, these pleasant substances, fat and sucrose, both activate very strongly. This is simply the time course of the bold response. Both activate this tertiary taste cortex region in the pregenual cingulate cortex. Now, it turns out that the pleasantness of fat texture is even represented in this pregenual cingulate cortex. Here it is in a coronal section. Here it is in a parasagittal one. So what we're looking at here is the pleasantness rating for the fat in the mouth, -2 unpleasant, +2 very pleasant, being given on every trial in a brain imaging experiment. And here's the bold signal. And we often see this. It's fascinating. There's a linear relation between the human subjective rating and the bold signal measured in MRI. And this is it for the pleasantness of fat being represented in orbitofrontal cortex here, or up here in pregenual cingulate cortex. Okay, so that's a little bit about the processing of reward. What about its modulation by cognition? If we show someone a food, something like this, when we give them an ambiguous stimulus, such as isovaleric acid, will the top-down cognitive input, for example, this picture versus this picture, make any difference to our brain's response to this stimulus? Now, isovaleric acid, for those of you who haven't smelled it, you might think of is like brie, if you're told it's brie. But if you're told it's soldier's socks, you might think it's soldier's socks. In other words, the cognitive label can make a big difference to how humans respond to it. The question is, where is all that implemented in the brain? Does the flavor reward representation go up into some language cognitive area, and it's all worked out there? Or does the cognition, what we tell someone in words, go down into your reward system? And it's the latter. So here's the experiment. Isovaleric acid delivered in an using an olfactometer into the nose, always the same on every trial. No pictures, but words, so that we're getting right to the top of cognition. And the words say, on one type of trial it's cheese, on another type of trial it's body odor. And what happens in the brain? Well, it turns out, here's our pregenual cingulate cortex, which is showing a bigger activation here if we've told them it's a reward, it's cheese, than if we've told them it's body odor, okay? And that's present in this region, pregenual cingulate, and coming down a bit towards orbitofrontal. But interestingly, it's also present in some places like ventral striatum. Now, we do a control experiment. We can give no odor, but just the top-down words saying it's cheese or it's body odor. And then we see the weak effect of the top-down input, but there's much less change of the signal, because really what's happening is the bottom-up smell is being modulated normally by the top-down input, you see? So this is just the control case, which shows you the top-down input, the weak effects of the word, and you get the big difference when you also have the smell present. So that's interesting. The conclusion is that cognitive effects, descriptions you might make in advertising, in describing your food, go right down into the bit of the brain that represents its pleasantness. Okay? So in a sense, what I'm really showing you here is that things like satiety signals, what we've been hearing about in the gut in the last few days, all the effects of gastric distension, chemosensing in the gut, come up into orbitofrontal cortex, and it's there, and probably in amygdala and hypothalamus, that you turn off reward neurons. And in the same brain region, you even get cognitive effects. So you see, I'm trying to synthesize the sort of stuff we've been hearing in the last few days. So that's top-down cognition. But what about top-down attention? So now I'm going to switch to taste. We deliver here taste monosodium glutamate on every trial. But on some trials, we say, remember and rate the pleasantness of what's going to be delivered here. And they do that after 30 seconds. Or remember and rate the intensity. So it sets the brain up into doing something else with respect to the identical MSG given into the mouth. And here's what happens. Let me say we image just about the onset of the taste delivery when the instruction is given. So what we find is that if we look at orbitofrontal cortex and pregenual cingulate, here's orbitofrontal, here's pregenual cingulate, we find that there's a bigger brain response if they're in the pleasantness condition than if they're in the intensity rating condition. And the same is true here for pregenual cingulate cortex. But again, you see this fascinating relationship. The bold signal is linearly related to the pleasantness of the food. You can read off, that is, from the brain signal, effectively, how pleasant the food is to the human. Okay, now let's switch to the other condition. This is anterior and mid-insular cortex responding to the taste of monosodium glutamate, but here, when you're paying attention to intensity, blue, you get a bigger bold signal than when you're paying attention to pleasantness. So the idea is you turn up the signal processing in your primary taste cortex when you're concerned with what it is, how strong it is, and you turn up your processing in the orbitofrontal cortex when you're interested in how pleasant it is. And here again we get a corresponding relation. The bold signal is linearly related in the primary taste cortex to the intensity rating given of the taste in the mouth. Okay, so selective attention seems to be able to select either a hedonic processing stream in the brain or a perceptual processing stream. Again, absolutely crucial when you're developing new foods to have these things under control, and there are possible implications for the control of food intake. Obviously, in terms of whether you draw attention to particular properties of food, its pleasantness or its quality, and from the previous experiment, the cognitive description that you use. Okay, so the next question is, when some people like particular stimuli, where is that liking represented that shows they're different to other people? Is it in the sensory processing system, or is it in the hedonic system? So we did an experiment with chocolate cravers. And chocolate cravers are identified using a standard food craving questionnaire, but also I can tell you that our chocolate cravers were validated by eating 200 grams of chocolate a week, whereas the non-cravers were eating something like 5 grams on average. Okay, so here's the experiment. We're delivering chocolate, liquid chocolate, into the mouth, and we're measuring the responses to the sight of the chocolate and the chocolate in the mouth, and so on. Here's what happens in the primary taste cortex. There's no difference, basically. Here's the primary taste cortex. There's no difference between the cravers and the non-cravers. So whatever it is, it's not early sensory processing that's separating chocolate cravers from non-cravers. Now, when we go through into pregenual cingulate or anterior cingulate cortex here, then we do see big differences. So chocolate in the mouth produces a much bigger effect here, and in the ventral striatum, in the cravers. The same is true for the sight of chocolate in, for example, the orbitofrontal cortex here, and bits of cingulate here, and bits of ventral striatum, a much bigger effect in the cravers and the non-cravers. So the idea is the effects of individual differences in making you like particular foods are being expressed in the hedonic system, things like orbitofrontal cortex pregenual, but not in the sensory analysis system. So understanding individual differences in brain responses to very pleasant foods in the bits of the brain that represent pleasantness may help us to understand the mechanisms that drive the liking for particular foods. So this leads me to make a slightly more general point. Each specific type of reward, taste, flavor, water, and so on, is represented by different neurons. And I've argued in Emotion and Decision-Making Explained that there are genes that specify the taste receptors to make different individuals like different tastes and rewards differently as part of natural selection and the way natural selection works. But if that's true, that means we're all going to be slightly different in the rewards that we find attractive. So looking for individual differences in sensitivity to food and using that to account for sometimes for differences in craving or obesity is likely to be important. So this is just a summary of orbitofrontal cortex neuroimaging, taste, olfaction, reward there, whole food reward, viscosity and fat representations, individual differences are reflected in places like orbitofrontal cortex. So this enables me to summarize this little bit of the talk, which is my sort of main sort of scientific contribution. I'm then going to have one or two slides that raise questions for the following discussion. So what I've said here is this is the system that in primates and humans appears to be implementing the reward value of these different foods. We bring together by convergence all these taste, olfactory, and visual inputs in places like orbitofrontal cortex and amygdala to compute their reward with modulation by these gut satiety signals. So the gut, all the digestive stuff that we've been hearing about, appears to be acting in orbitofrontal cortex, but not before, not here, nor in the primary piriform cortex, nor, as we've shown, in inferior temporal visual cortex. And then these reward representations can be biased by top-down cognitive or attentional control. And then we go from these bits of the system, this is the reward tier, onto another tier up here, which I'm not going to talk about today, where we then take decisions between rewards. So once you've computed that something's nice, you then take a decision about what you're going to do about it. And that is, interestingly, slightly probabilistic because of the way it's implemented in the brain. But that's what the book is about, emotion and decision-making. But I don't have time to go into the decision-making bit today. Right. So now I'm moving from the sort of synthesis I wanted to give you of what we've been hearing about in this meeting to introduce then and raise some thoughts for the following final discussion. So the first thing is obesity. Could obesity be produced by overstimulation of the food reward system in the brain? And in relation to that, there appear to be many factors, and any one of those factors could potentially tend to cause obesity. So to control obesity, you may need to pay attention to every one of these factors. And let's just run through them briefly. So we talked about endocrine and genetic factors. They're not very important in most obesity. They account, at least on current evidence, for just a small proportion of obesity, such as people with defects in the leptin system. Externality. So Schachter did some work originally that obese people may be more reactive to the sensory properties of foods. And what I showed you today is there are big individual differences in the reward system in sensitivity, and I gave you an evolutionary argument that that may be very important. So even developing food for different markets may be important. This factor of palatability, food companies have produced wonderful foods in terms of flavor, but that, the argument is, overrides our pre-existing satiety systems, sheer palatability. What's the solution? So one solution is to continue to produce highly palatable food, but to make it healthier. In other words, it's a challenge. It's not a problem. Variety, sensory-specific satiety, is a factor that can contribute to overeating, as we've seen. The meal pattern is quite different in most people who are obese. Portion size tends to be bigger. Energy density, eating rate may be too fast. So all of those things, again, need to be taken into account if we're to understand obesity. Meal concentration I've mentioned. Stress-induced eating, another factor that I raised in relation to the intermittency that we heard about earlier. And in general, the regulation to internal signals is very poor in humans. It takes a long time. In other words, these gut signals that we have don't switch us off very quickly when we get satiety signals in our gut. It can take something like, in humans, 1 or 2 weeks to adjust to a change in energy density of food. So in other words, humans are dominated by these sensory inputs. There's one other important factor just to raise for discussion. I've been talking about what you might call a pleasure system, which computes how nice something is and gives rise to goal-directed action. We, of course, as humans, have an explicit reasoning system. So we can decide that we don't want to eat all of this very nice ice cream because it might make us obese. But notice that these systems have different goals. The goal of the reasoning system can be to produce long-term optimal behavior. The goal of these gene-specified reward systems can be to produce behavior that's good for your genes. They'll be more short-term. So these two systems may be in competition. Who's going to decide between them? Again, that may be different between different individuals. Some individuals may be more susceptible to advice and operation of the cognitive control system. Others may be more dominated and more impulsive by their gene-specified reward system. Right. So what could we do in the future to try to sort of analyze what's happening? So there are many factors that could produce overeating. We would have to control all of them. Are the brains of individuals who tend to become obese more sensitive to rewards? Well, one possibility that I've suggested already is that we could do very large-scale imaging studies and then post hoc have a look in relation to BMI, whether there are differences in certain brain systems, and which questionnaires, be they addictive or not, might correlate with that. And are they also less sensitive to the satiating property of food? That would also be extremely interesting. Can we predict weight gain, say, over 1 year from the sensitivity of the food reward system in the brain? So all of these are fairly open questions. And I'd emphasize that decision-making itself is probabilistic, so that we have to allow for the fact that humans may do different things on different trials. Now, to get back to what we heard yesterday about satiety signals from the digestive tract. Is gut feedback nutrient specific? We raised that question yesterday, and there's some evidence that the conditioning effect for appetite is, so for example, does MSG in the gut reduce the pleasantness of the flavor of protein? Or does glucose in the gut reduce the pleasantness of sweet taste? We don't know much about that yet. But notice that there's also this claim, which I mentioned yesterday, that MSG in the gut can have a super strong satiating effect and can help to reduce dietary obesity. That needs to be followed up. Notice that the effects of food in the gut can do many different things, and I hope that the people who come up onto the platform in a few minutes may be able to take these as questions. Unconditioned satiety, conditioned appetite, conditioned satiety. So far, we've heard about conditioned appetite from them. And then lastly, obesity, addiction, and the mismatch hypothesis. So the mismatch hypothesis seems a reasonable one, and whether we want to go on to talk about addiction may be influenced by a number of things. But of course, the control of eating is different from addiction in that we have satiety signals for feeding, including sensory-specific satiety. So at least technically, there is quite a difference. Nevertheless, these very pleasant foods may produce so much reward that in some cases dopamine's released, and it turns out that dopamine has a strong projection into your orbitofrontal cortex, where it could influence reward. And then I've got this point about the large-scale database. So those last few slides are just intended to introduce some of the topics that we might want to discuss. And overall, then, what I've tried to do, and this is also what I tried to do in that book, is to give you a framework for understanding how all of these factors come together in certain reward bits of the brain to produce our behavior. I'll stop there.